Um, so my name is Damon Harity. I'm a wildlife program manager at Sky on Alliance, and we're joined by a number of our staff. Uh, and the highlight of today is Sarah, a PhD candidate from University of Tennessee, Knoxville. She's a incredibly dynamic speaker. She's doing research on Sky Island complexes and how they can be natural laboratories for studying the evolutionary impacts of climate change. And she looks at this kind of globally. She does a review of these systems around the world, and then also does local field experiments here in the United States. But we're in for a treat. I've seen her present. She's great, super knowledgeable, very enthusiastic, and excited to hear what she has to say. So I'll turn it over to Sarah.
Thanks so much, Eamon. So I'm going to share my screen and let's just make sure that we have the right view. All right. And so do you all see the presentation?
We do. Yeah, that looks perfect.
Awesome. Okay. Well, again, thank you, Eamon. I really appreciate that very kind introduction. Again, my name is Sarah Love. I'm a fifth year PhD candidate in the Schweitzer and Bailey labs at UT. Today, I'll be talking to you about Sky Islands as natural laboratories for studying the eco-evolutionary responses to climate change. First, I have to thank the people that made this work possible. So my advisors, Joe Bailey and Jen Schweitzer, fellow graduate students, Ashlyn Horde and Caitlin Barnes, and our GPS man and rockhound, Abel Bailey. This network extends much past what is on this screen, but I also want to thank my department of EEB at UT and my GATE fellowship at Oak Ridge, as well as all of my collaborators, support, and committee members. So jumping in, the premise of this talk is to understand how adaptive variation to previous environmental change can help us predict how extant systems will respond to ongoing climate change. And the problem is there is a scarcity of natural systems to examine these questions. Previously, we've used these methods on the bottom of the screen here. We've used paleoecology or using fossilized organisms and historical climate to study climate change, but these are all observational. There's no genetic variation in them, and they're not widely reliable for many species. We've used space for time substitutions across latitudinal or elevational gradients to study responses to different climates, but these often have lots of confounding variables across these gradients. There's limited replication in the studies, and they're limited to species with a notable elevation or latitudinal range. Experimental manipulations have been widely used to study climate change, but these are often short-term single-factor manipulations with highly artificial conditions, and they can kind of produce false adaptive responses because of this. And finally, we have resurrection studies where we use preserved specimens from hundreds of years ago using seed banks. And so these are restricted to plants. They may not reflect contemporary genetic diversity, and they're limited in scale. So each of these methods has provided valuable insight across ecological disciplines. But today, because of the limitations and because we are in dire need to understand climate change better, I want to talk to you about an additional approach to meeting the above objective. Introducing Sky Islands, many of us are very intimately familiar with these systems, but for those of you who are not, the Sky Island is an isolated mountain peak surrounded by a lowland that serves as a barrier to dispersal, commonly desert. As seen on the left-hand side here, those of you at the Sky Island Alliance will know that this is Mount Lemmon. This is right outside of Tucson, Arizona. You can see in this screen grab from Google Earth here that this mountain peak is surrounded by the Sonoran Desert. On the right-hand side, this is a beautiful map by the Sky Island Alliance where you can see Mount Lemmon right here in Arizona. Can you all see my cursor, by the way? I don't want to continue assuming that you can.
Yeah, we were able to
Okay,
see that, Sarah.
awesome. Okay. These sky islands began forming around 200 million years ago, mostly through uplift through tectonic plate collision, also through volcanic activity, or through the erosion of previous mountain ranges. And once these mountain peaks were formed, they were started to colonize. So at the top here, we have the initial colonization as represented by the orange stars. Over time, with climate change, deep climate change, these populations allopatrically diverge. Those of you who are not evolutionarily inclined, allopatric divergence is divergence due to a physical barrier. So because of this isolation, they began to become genetically unique. With the Pleistocene, with glaciers, these populations were moved downslope where they became panmictic. This is another word that I'll use a lot, which essentially means one large population that's able to breed. After the glaciers retreated, these populations tracked that cool climate, moving back upslope, where they have remained today, diverging again allopatrically. And this is exacerbated, as we found in our literature review, by modern climate change. We also found in our literature review that these sky islands are not just isolated, pun intended, to the southwestern United States and the commonly studied Madrean archipelago that the Sky Island Alliance is very familiar with. They are also globally distributed. So we have major sky island complexes in the Madrean, Rocky Mountain, and Great Basin archipelagos in the western United States and northern Mexico. We have the Ethiopian highlands and East African arc and the Cameroon line in Africa. We have the Annamite range in Asia and the Western Ghats in India, and the Pantapuy range across Venezuela, Guyana, and Brazil. And these studies span diverse taxa. So though the Sky Islands obviously are home to many distinct organisms, the majority of those that are studied in the literature are plants and invertebrates. Though a few studies did examine Sky Island trends on mollusks or fungi or reptiles, for example. And so overall, the pattern that we found in our literature review is that climate-driven Sky Island isolation results in past, up here, and present population divergences. The notable findings are that this Sky Island isolation results in reduced genetic variation, habitat reduction, smaller population sizes, lineage formation through allopatric divergence, morphological divergence that tracks this, speciation, and ongoing selection through climate change. So let's jump into some of these examples now that we have the background out of the way. I love this example because this highlights how Sky Island isolation does not just influence sessile species or those species like plants that are not able to move. There is also notable effect of isolation on highly mobile species such as birds. So here we have the Nilgiri blue robin on the left-hand side. The study was in the Indian Western Ghats by Robin et al. 2010. And on the right-hand side, we have Lupinus alopicuroides in the Northern Andes as studied by Vasquez et al. In 2016. And what they found using these comparative studies across Sky Island complexes, they looked at the convergent genes associated with relic population divergences to determine these large-scale evolutionary strategies for persistence, such as interspecific hybridization, or here, population-level diversification. On the left-hand side, what they found was that the Sky Island populations are significantly genetically distinct from one another. Again, this is in the Nilgiri blue robin, highly mobile species. Notably, this divergence, so again, this is the Indian Western Ghats, these northern and most southern species are genetically distinct enough to warrant them as different taxonomic species. So speciation is happening in this system. And the study found that this is not just a result of the distance from and degree of habitat change between the refugia, it's also through modern climate change. And so they found that though these populations initially started diverging in the Pleistocene, and the lack of gene flow prevented or promoted this divergence, these population differences have been maintained due to anthropogenic stressors such as climate change. And the same story is true on the right-hand side in Lupinus alopicuroides, where there is extremely low genetic variation within each of the sky islands, as seen in the little triangles here. But there is extremely high genetic variation across this the study sites examined. And this is pretty common. This is a pretty common trend in the literature review where we have really genetically homogeneous populations when you're looking at single sites, but then when you zoom out, it's incredibly diverse because each site is distinct. But in our literature review, we did not just look at genetic variation. We were very curious what was happening at the phenotypic level or the trait level. Most of these studies, if they reported, that's the key there, because they didn't all report phenotypic change. If they reported phenotypic change, it was mostly to what we call body or body size, shape, color, etc. Though some studies reported differences in immunity, metabolism, or thermal tolerance in the Sky Island populations. So to jump into a couple of these examples, one of my favorites, and I try to not be too biased, but it's really hard when you're studying some really fascinating areas. This is one of my favorite examples. This is Huxley and Spasojevic 2021. They did a community level analysis. So they used LIDAR and field-based sampling to look at overall community changes on 7 different sky islands in Colorado. And they looked at these changes in 2008 and 2020. And so they had 12 years of climate change where they had the associated climatic differences within their study. What they found is that over these 12 years, there was a shift towards increased species richness and phylogenetic diversity. Specifically, in the trait in their traits, they shifted towards increased stress-tolerant leaf traits. So for those of my plant people out there, what they found was that there was lower SLA or specific leaf area, so they're smaller leaves, and they had higher leaf dry matter content, so they're denser, smaller leaves. And for my really nerdy plant people out there, they had a decrease in functional dispersion of SLA. So overall, there's convergence on all of these sites towards those dense, small leaves. And what this study found was that these effects were mediated by island size, so how big the refugial area was, and geographic isolation. So the larger islands had greater environmental heterogeneity and larger communities, and the smaller islands saw more dramatic effects of the 12 years of climate change phenotypically. We also see some interesting results for traits for persistence on sky islands. So this example is by Olin et al. 2000. They looked at Pinus balfourania or foxtail pine in California, USA. What they found is that in the more stressed climatically sky island sites, the Pinus balfourania had adapted to serpentine soil. So serpentine soil is this really uncommon soil type as a product of weathered ultramafic rock. This essentially means that there's very low levels of nitrogen, phosphorus, and potassium. And this soil type is very inhospitable to many terrestrial plants. And so in these more stressed sites, the foxtail pine, which is some of the longest lived species on the planet, have adapted to survive on this very rare soil type in comparison to the less stressed sky islands where they're on this granitic soil. So these traits for persistence are important to population outcomes in these stressful environments. And overall, the Sky Island differential adaptation to stressful environments combined with population bottlenecks can lead to speciation. And in fact, we find that in 25% of the studies, there was speciation or an indication of speciation reported. So in this example, this is a Delphinium species. There are 3 main species examined here in the East African Rift system. These 2 graphs combined show that, so the colors are indicative of where they come from geographically. So the colors on the left-hand side here, we'll just focus on the blue, yellow, and red. They map to this, the geographic sampling sites on the right-hand side here. So this is a PCA, or principal coordinates analysis. And this essentially shows that these 3 species are genetically very distinct from one another. They cluster together, so they're similar to each other, but they're very different from one another. And interestingly, this maps to where they come from and which sky island they come from geographically. And again, this is one of those, like, it's hard to not pick favorite studies, but this is a really, I have to say, this is one of my favorite studies. Because in their conclusions, now they didn't test these hypotheses, but in their conclusions, they started to speculate about why these phenotypic patterns would be present. So not only are they genetically distinct, as we see in this PCA, but there's also very significant phenotypic differences. This portion here is the sepal of the flower. And so they asked, why would Delphinium macrocentrum in blue here have this incredibly long sepal in comparison to Delphinium dasycolon in red here, where it has a really tiny sepal? I'm going to take a pause and just put on your ecology caps. Why could this be? I'll give you the answer, or at least the answer that they gave in their conclusions. Maybe it's due to pollinators. So this Delphinium macrocentrum, again, this is the more central site. The pollinator that is present on these sky islands is the hawk moth, which has an incredibly long pronotum. In comparison to those northern and southern sites where Delphinium dasycolon exists and is pollinated by bumblebees. So I think that this is a really interesting example. And though it's quite rare in the literature to look at community level effects in the Sky Island sphere, this shows that these divergent Sky Island communities can influence speciation and really is an area that needs more attention. And this speciation doesn't just happen because of geographic isolation. It is also directly influenced by habitat contraction driven by climate change and human encroachment. So this example is Bolitoglossa orispia from a single sky island in Honduras. So this is a newly described species. it is physiologically distinct and reproductively isolated from all other salamanders in the nearby region. Its range is a single hectare of forest remaining on top of this single mountain. It is now the most critically endangered salamander in Honduras. And with human encroachment, you can kind of see some of these roads and buildings. Its habitat is shrinking dramatically. So we just went through the background of how Sky Islands were formed, how populations colonized. Then we talked about some Sky Island effects and notable patterns, both at the population and a little bit at the community level. Now I want to move into the comparative approach, because these terrestrial islands don't just exist in isolation. Again, pun intended. They exist next to large connected mountain chains often, whereby the different climates and habitat connectivity on mountain chains results in very different landscapes. So again, we have this example of Mount Lemmon, and to the northeast of it is the Mogollon Rim, which is this large series of mountain peaks that have high elevation habitat that allows the populations to remain connected. In our literature review, overall, we found that in comparison to Sky Islands, mountain chain populations show higher genetic variation and gene flow. How does this happen? This is the same schematic that we reviewed earlier, where we have the initial formation all the way down to what's happening in modern day. And in comparison to what's going on the right-hand side on Sky Islands, on the left-hand side with the mountain chains, we see the same processes essentially happening from the formation. They diverge allopatrically. There's panmixia when the glaciers are present, then upslope retreat when they retreat. But in modern times, today, there isn't distinct allopatric divergence or divergence because of a physical barrier. Because there is this connected series of mountain peaks where the high elevation habitat is more connected, the populations are allowed to, they have higher gene flow, which maintains higher genetic diversity in comparison to the Sky Islands. But in the literature review that we conducted, and we reviewed over 800 papers, only 0.05% of the literature had a comparison to any kind of mountain chain if they examined Sky Islands. So a very, very small fraction. But those that did examine the Sky Island mountain chain comparison showed some really interesting effects. So on the left-hand side, we have the 6 studies that were used. We have many different taxonomic groups represented, from black bear, Ursus americanus, Pinus balfourania, Sclerobonus robustus, Tamias species, Neophasia species, and Strix occidentalis lucida. They're all in the southwestern and western United States as well. But they show some really interesting patterns. So in support of our hypotheses for the Sky Island mountain chain comparison, these studies showed that the Sky Islands exhibit extremely low levels of gene flow, low genetic diversity, smaller populations, and selection-driven habitat specialization in comparison to the mountain chains, where there is high gene flow, they are panmictic, and they're more likely to be habitat generalists. So to dive into 2 of these examples, on the left-hand side, we have Neophasia species, which is the pine white butterfly in the southwestern United States. And then in this map, you can see that they used the Madrean Archipelago as their Sky Island region. And above that, they examined the mountain chain region from the Kaibab Plateau, Mogollon Rim, and the White Mountains. Interestingly, the mountain chain populations were reported as panmictic with minimal population genetic substructuring. So they were essentially what they found was that the Kaibab Plateau, some of these populations from the north up here, were still able to migrate down to the White Mountains and reproduce with some of the populations in the south. In comparison, the Sky Islands had significantly diverged in a hierarchically clustered isolation by distance pattern. So what this means is that the Sky Islands, not only are they distinct from one another, as you go further away from the Sky Island, you are more likely to be even more genetically distinct. And so even though, the Pinalino Mountains and the Chiricahua Mountains, this green band here indicates like medium elevation. In theory, it is a highly mobile species. They could migrate to the south to reproduce with the Chiricahua population. They are not. And if they do, it's very rare. And on the right-hand side, the Strix occidentalis lucida, the same pattern was reported. There's not as pretty of a map. They studied a similar area, but they essentially found that the mountain chain populations were panmictic and the Sky Island populations had significantly diverged. And the reason that I picked these two studies to use as an example is because, again, these are highly mobile species. So one may posit that in theory, the Sky Island populations or Sky Island isolation should not have such a significant effect in a mobile species, but we're seeing because of where their habitat is, where their nesting grounds are, where they're going to reproduce, Sky Island isolation does not discriminate in between vagile and sessile species. It affects everything, even a smaller mobile species like this Neophasia butterfly versus the larger owl. Also, from the comparative studies that we examined, this is a study by Bell et al. 2021, where their focal species was a species of chipmunk under the genus Tamias in the northwestern United States. This is the yellow pine chipmunk. What they found was that on the Sky Islands, these chipmunks were colonizing this unique habitat of Pinus albicollis, or white bark pine, in comparison to in the mountain chains, where they're more habitat generalists. And this is notable because Pinus albicollis habitat is under significant decline because of anthropogenic stressors. And so what this suggests is that the Sky Island populations of the Tamia species of the chipmunk could be at risk for disassembly, and these Sky Islands writ large at risk for community disassembly, whereby the declining populations of Pinus albicollis exacerbates the decline of the chipmunk population. So we've learned quite a lot from these 6 studies that use the Sky Island mountain chain comparison. But all of these studies are population level observational field-based studies. They are molecular in nature, looking at non-coding regions of the genome, and they don't use quantitative trait change to examine the effect of climate change. And so we can build on this work using common garden studies with modern climate taken into consideration. So for one of the chapters of my dissertation, I worked to analyze a common garden study that took place across the natural range of Populus angustifolia to test the effect of the Sky Island mountain chain comparison, specifically how the climatic differences influence quantitative traits. First, we wanted to make sure that the climates were different and test this hypothesis. And so we extracted climatic data from all of these sites. Again, we have the Sky Islands in red, orange, and the mountain chains in blue. And what we found is that here on the left-hand side, the Sky Islands had significantly higher mean annual temperature. And on the right-hand side, significantly lower mean annual precipitation than the mountain chains. So they're hotter and drier. We also looked at these climatic differences across the last 120 years, though they most likely extend further past this. These were just the data available at the time. So what we found is that the Sky Islands have been hotter and drier than mountain chains for at least the last 120 years. Again, as I said, we use Populus angustifolia, James, or narrowleaf cottonwood. Many of you out west will know that this is a dominant native in the riparian corridors across the western United States. It has an immense pool of genomic resources. It was the first tree to have its genome sequenced. It reaches sexual maturity very quickly, which allows us to look at the traits that we want pretty quickly after planting. And they're easily propagated by clonal cuttings. And so we can go out into the field, take some terminal branch cuttings, and propagate them in a greenhouse. We looked at two main traits for this study, which is above ground biomass, which represents carbon sequestration and may influence overall ecosystem productivity, as well as cloning or asexual vegetative reproduction, as this allows a genet to spatially scavenge for resources under the soil and across the landscape. And cloning allows trees to persist in environments post-perturbation or under stress because they can clone from even damaged standing parts of trees. Our overarching hypothesis for this study was that the Sky Island populations have diverged from mountain chain populations in reproduction and productivity traits. Again, that cloning and biomass. We find support for this hypothesis, whereby on the left-hand side, we found that Sky Island trees cloned more and more often than mountain chain counterparts, and they produce significantly more above ground biomass than their mountain chain counterparts. So this suggests to us that these trees are more resource use efficient. The Sky Island trees are more resource use efficient when grown under common conditions. But these trees don't just occur in isolation. They occur with their microbiome and they influence resources on the landscape. So going forward, this is a little teaser into where I've been working most recently. We understand that Populus angustifolia has an extended phenotype. And so because it's this dominant tree out west, what happens at the population level very quickly and profoundly influences what happens at the ecosystem level. And so we just saw that at the population level, we have differences in cloning and biomass. At the community level, we'll be looking at differences in soil traits from immediately underneath the trees. And at the ecosystem level, we're actually looking at soil nutrients, but here I have some other species represented because it's a little hard to represent soil nutrients graphically. But we know that changes in nutrients in an ecosystem influences the entire ecosystem. And so our analyses for these are as follows. So on the left-hand side, we have this population level divergence. In the middle here, from these soil bacterial communities underneath Sky Island and mountain chain trees from the field, we see that their communities have significantly diverged in composition, as represented by this PCOA. And then at the ecosystem level, we see that the Sky Island soils have significantly higher C/N ratios than the mountain chain counterparts. So this indicates to us that the Sky Island mountain chain effect extends to alter communities and scales to change nutrient ratios at the ecosystem level. And these are interesting observations, preliminary analyses that can serve as jumping off points for additional work. So where I am and where I'm going now, I'm wrapping up some analyses for this full factorial reciprocal transplant of trees and soils across the entire range, the majority of the range of Populus angustifolia, to determine the mechanisms behind the Sky Island mountain chain effect. And though I was very tempted to rush and put some of my new analyses here, I did not do that because we're still digesting the data. And so for now, I will just say there's interesting stuff happening. And maybe in a year, we can check back, do this again, and I'll report on what we found. So overall, what we've talked about today is that Sky Islands face the compounding effects of isolation, limited habitat, and long-term climatic differences, which has led to patterns of convergence in the southwestern United States, as well as strong ongoing selection, lineage formation, and genetic diversity across the globe. And this genetic homogenization and highly specialized Sky Island populations suggest that they may be under significant risk under increasing climatic stresses and geographic isolation. But there's also an additional story. This past lineage formation and current local adaptation plays substantial roles in driving population and community composition. And this is likely very important, as we've seen, to habitat specialization, the evolution of novel phenotypes, and the evolution of novel intraspecific interactions that can be used for preparing for the future under climate change. And so to employ this system more broadly, this is a call to action. A lot of you in the audience work in Sky Islands, and so I would love to talk with you further about this work and how you could employ some of this work, the comparative work, in your own systems. But as we've reviewed, the Sky Island mountain chain comparison is powerful in comparison to those first systems that we talked about at the beginning of the presentation, because the mountain chains and Sky Islands have evolved under the same evolutionary timeframes, but very different climatic regimes. And so these, we know, are systems to examine evolution, but we are now suggesting and seeing evidence for them being model systems to examine climate change moving forward. Some areas that we think should be the next era of Sky Island and Sky Island mountain chain research are global comparative studies, experimental manipulations, especially common garden studies, where we can tease apart maternal effects and phenotypic plasticity, as well as genes to ecosystems work. Overall, we can use the system to see how ecosystems have responded to climate change in the past to understand how extant ecosystems will respond in the future. That is it for me today. I really appreciate your attendance, and I would be happy to take questions now. I also have my email on the left, and my Google Scholar on in this QR code. But I would be happy if you're ever, if the papers are behind a paywall, just email me and I'll send you PDF copies. Our annual review was published earlier this month, and we just had a review of the ways that we study climate change published in PLOS Climate 2 days ago. So check it out, and I'll take questions now.
Wow, thank you so much, Sarah. That was really fascinating. Feel free, everyone, to unmute and ask questions or post them in the chat, and we'll try to make sure everything gets answered. I think Joe Cicero has a couple questions for you, Sarah, and I'll let Joe either.
Can she hear me right now?
Yes, you can.
Oh, wonderful. Hi, Sarah. Thank you very much for this seminar. It answers a lot of my questions about the formation of the Sky Islands and the pop comet ecology involved. So I really appreciate that. Can you go to the slide with neophasia, please?
Yes. Let's see.
Yes, that's the one. I interpret this female, the orange one, as a member of a relictual mimicry ring, because the helicones in the dennated south in Mexico are models for Batesian mimicry as well as Mullerian mimicry. And this one appears to be, it's in the Huachucas, and it appears to retain that configuration, whereas the other ones, Neophasia, Menapia, and so forth, do not. Have you added that into your calculus, that this thing is attached to the tropical and the neotropics because of that color pattern, relictual mimicry color pattern? Go ahead.
So I believe that, so the white is Neophasia menaipi, and the orange is...
Oh, I see what you did. Okay, so I thought this is the male. The male of Neophasia terludii is white also.
So I know that in this study, they actually used two different Neophasia species.
I see.
There was some overlap in their sites. And so some of the Turlui were in, both the Sky Islands and the mountain chains. I'm not as familiar with what you are talking about now. So what you're suggesting is that they also extend into the tropics?
Well, no, that at least the color pattern was derived from the tropics, because it used to be, or it is extant as a model for mimicry. And this thing, at least its evolutionary origins were down there. And this is the one and only one that made it up into the United States with that kind of color pattern. So anyways, it should be into your calculus, I would think.
Yeah, that's interesting. This is not my study.
I see. Yeah, sure.
Yeah, but I would be happy I can send you the PDF of this study, and we can talk a little bit more about your hypotheses around what's going on here.
That'd be nice. Please do.
Okay, great.
Can you go to the, I have two more. Can you go to the delphinium and the sphingid?
Yes. Let's see. Yeah, if you want to maybe write your email in the chat, I can get that to you.
Yeah, Eamon and I are good friends. We'll get in touch.
Okay.
That's real interesting stuff. This is extremely interesting. I've noticed these differences that you describe, and you attribute this to pollinators, which makes a lot of sense now. This in number C is a nectiferous spur. There's got to be a nectar gland at the tip of it that the sphingid is going for, right?
Possibly.
Well, I would think so. Otherwise, well, that's the reason why the proboscis of the sphinges is so long, to reach deep down into these nectiferous spurs. And with your bombus over here in A, or a D, that makes a lot of sense, because it's a narrow pocket, a short pocket that the bombus mouth parts can reach, where that should have a nectiferous gland also. So is that included in your calculus?
This is also not my study.
Right, exactly. You're taking secondary literature or primary literature and collating it into your study. No complaints. It's just I'm pointing out some things that I know about that should be figured into your calculus. Because that's what it's all about. The whole thing of insect mouthparts, elongation or short mouthparts, has to do with reaching that nectivorous gland so that its face smashes into the pollen trying to get to it.
Yeah, I don't remember reading anything in this study that directly mentioned that.
That's my point, because it should be, because you need to go to another literature, and that would be the monographs on delphinium that I imagine show even transmission electron microscopy of that nectivorous gland to demonstrate that it's there, and that's what the insects are going after. So
yeah, they so they were just postulating at the end of their study, just in their conclusion. They, found these genetic divergences and the physiological divergences, and then basically at the end said, oh, and the pollinators are very different, and don't, the hawkbill moth does not exist on the, in these northern and southern and western sites.
Yes,
is present in the center.
Well, there's hawk moths up there, but they don't go after the disc, and they probably have proboscis that aren't as long as the one that is co-adapted to that. Anyway, that answers my question. It's another line of literature that needs to be put into this calculus. And lastly, before I don't want to consume any more of your time, let's go to the panmixtic, the mountain range pictures that you showed. Al Patrick. Yes. Okay. So we have an icon species here in our Sky Islands down in southern Arizona that is extremely important in this kind of study, because it shows an exception. The species that you've shown us fit your models, but what about the exceptions? And if you study exceptions long enough, you'll find that they're the norm. Maybe, I don't know that. You have to look for exceptions to your model also. And we have, as an example, an icon scarab down in our Sky Islands. It's Chrysina gloriosa. It's a beautiful scarab, green with silver stripes. All the insect collectors go after it. It occurs clear up in all the mountain ranges, clear up to Payson, with very little change that I've noticed. Whereas its conspecifics, there's two right there in the same mountains, do not. And one of them, LeContii, is confined to the higher altitudes, which figures into what you were talking about also. So I guess my point is to speak about the exceptions as well as those that fit your models. Okay, I don't want to take any more of your time. I imagine there's a lot of other questions.
Well, I'm eager to talk with you. So what you're saying is that you don't find one in a mountain chain where you would expect it to be. Did I understand that correctly?
No, well, I'm saying that there's three species that we have here in the Huachucas and the Santa Ritas and the Chiricahuas that are very similar to each other. There's another one in Texas we won't get to. But one of those three, that'd be Gloriosa, has made it all the way up to Payson that I know of, with very little change. I don't know if there's any change at all. And so that's an exception to the things that you're talking about. So it's in all the mountains that I know of.
Sky Island species that's able to migrate to...
Or be blown, yes. Or it got... with the separation of these mountain ranges, as you described them, it stayed with them, I guess. The other ones did not. I don't know. It's over my head. I don't study what you're talking about, but it's a new leaf for me to think about these insects that I know of, and it's a good example too. I imagine there's other examples of species that extend their ranges clear across the board, whereas their conspecifics have stayed behind. So anyway.
Interesting. I would love to hear more about that. I'm curious if you have any manuscripts or data on that.
Yeah, sure. We can correspond an email. Let me throw the ball back to Eamon.
Thank you so much for your questions.
You're very welcome.
Yeah, thank you so much, Joe. We've got a few more minutes of time. So if anyone has questions, feel free to chime in or put them in the chat. I did a quick question, Sarah, from my own. At the end, you made a point about how Sky Island populations might have, because of their lineage, because of their diversification, and because of their sort of history under different climatic regimes, they might be. Were you suggesting that they might have some of the genetic tools, so to speak, to adapt to changes? Or did I misunderstand your point there?
Yeah. Yeah. No, you're exactly right. I mean, it's either or,
Yeah.
right? As Joe was talking about, like we have to study both the exceptions and the rules, or however you want to say it. On one hand, they could just not be doing well. But what we're suggesting and what I'm looking into is, well, actually, they've evolved for 15,000 plus years under these incredibly stressful climates, and so could we use them for assisted migration, for example, to these mountain chain populations that will face increasingly hot and dry or unpredictable climates in the future. And so what we showed in our, common garden study was that when these populations are brought into common conditions, and given a regular watering schedule, they produce significantly more clones and above ground biomass, which is not what we were expecting, because it's typically either or. They're typically, phenotypic trade-offs between sexual or asexual reproduction and growth. And that's not what we found. We found that they're just able to utilize the resources and do both much better. Now, we don't know exactly how that would play out in the field, but that's one of the researchers that I'm talking to for a upcoming potential postdoc does more of this applied conservation work, and so we've been talking about ways to test how these systems and how these questions would play out in the field. But in theory, they should have genes and traits that could help them persist in stressful environments. Yeah.
Yeah, thank you so much. We had a comment come in that it seems that sky islands are more vulnerable to the climate crisis and species extinction, which is, I'll let you kind of build on that or respond to that, but it's related to what you're saying.
Yeah. Oh, yeah. So that last photo that I showed, this was, we're driving past Moab. This is Pack Creek, if any of you are familiar. And I was with Ashlyn Horde, who I mentioned at the beginning. She studies fire and she studies hybridization in the same system. And so we're looking for these fire sites. We found this fire site, and all of this at the bottom here, first of all of this at the top is burned Populus angustifolia. And all of this at the bottom is not willow. It is narrowleaf cottonwood, which, my advisors have worked in the system for the last 2.5 decades and had never seen anything like this. And so, yeah, I mean, they're being charred, and though they're prolifically regenerating, this is not going to stand a chance if another fire comes through here. So essentially we're like, what this place that we are right now is, if there are genomic resources, we better use them soon.
Sure.
We may be losing them.
Right.
Yeah. Tim, do you have other thoughts on that?
No, except that I think it's really important for all of us environmentalists to publicize this and to send, to ring the alarm and talk about. I think many of us realize about the species extinction, but I don't know how much people understand how serious this is.
Mm-hmm. Yeah. Yeah, it's pretty concerning when, again, your advisors have studied this for 25 years and some of the sites that they've gone to, say, like right outside of Las Vegas, don't exist anymore. Or in this system, and this is a can of worms, but maybe they're just maintained by hybrids, which is a really interesting component of the populous system. Yeah.
Well, we are just about out of time. If there's one last question or comment or anything, we've got maybe time for one, but just want to thank you, Sarah, for taking the time today to speak with us, present your fascinating work. We're really honored to have you today. And yeah, we look forward to hearing more from your exciting work where you're going in the future.
Thank you. Yeah.
And just so everyone knows, this will be recorded and shared. If you have friends or family that might be interested in seeing this, we'll quickly share the link so anyone can watch this, a recording of this presentation. But
yeah, thank you so much, Eamon. Thank you, everyone from the Sky Island Alliance. The work that you're doing it does not go unseen. So I really appreciate the invitation and opportunity to present to your brilliant scientists and community today.
well, yeah, thank you very much. Everyone have a great day. We're so glad you could join us, and feel free to reach out and I can put you in touch with Sarah if you have follow-up questions.
Yeah.